in all the therapies we have, Rebif, Avonex, Betaferon, glatiramer acetate, and lately I haven't included that on the slide, Rebif, they have shown that if you start extremely early, you see a delay in the second relapse, and one study already showed that you see, and that was Betaferon in the benefit trial, that you have an impact on disease progression within 3 years. That has not been expected at that time. The order of magnitude, you see that in this slide, is around the 50%, so the likelihood to reduce the risk to convert into clinically definitive MS means to experience a second clinical relapse is approximately 50%. There is a bit of a dose dependency. That means there is a direct dose relationship. If you directly compare, for example, subcutaneous interferons, as has been done in the reflex study. With regard to the relapsing remitting population, I don't really go through all the studies. There is plenty of them. What you can see here, if you take such a slide, which is always a bit comparing apples and pies, to show the relapse reduction rates in the pivotal trials. And what you see is that the ABCR drugs are within the 30 to 35% range of reducing the annual relapse rates. And from the latest trials we have seen, we have much larger reductions in the relapse rates, which has to do both with higher efficacy of these drugs, but also with changing populations and lower event rates at all. The big question that we are now starting to answer, and it's still not clear, it's hard to read because the conversion from Mac to PC somehow killed my transparency in this slide, is the long-term benefits. So the question is, what is the real long-term benefit of the interference? And you know that all the approval studies and the classical double-blind controlled studies are for 2 years, maximum 3 years, but thereafter you have to unblind patients and you cannot do that in an uncontrolled fashion. But nonetheless, people get their treatments for definitely more than 2 to 3 years in the ideal case. And that is actually cumulative data which I picked now from the preparation interferon beta 1b, that have in all different MS segments now data on long-term benefits. So you see, if you start very early, then you have an advantage of very early intervention versus delayed intervention. You have a certain impact also on cognitive features, which is measured by the PASAT. This is a very interesting outcome that nobody would have really expected, but it's published and it's now a fact that you could say people that have taken continuously interference throughout their disease course, they have an influence on a very robust endpoint, which is mortality in multiple sclerosis, which is not the endpoint we are really trying to address in the very beginning, but nonetheless, obviously, there seem to be factors that are influenced. And there is also influence on the development of persistent tissue destruction that you can measure by MRI imaging, which is given here by the number of black holes. This is a study that is often cited because people want to know what is a very robust endpoint with regard to the long-term treatment efficacy of the interference, and this is conversion to secondary progression. What has been done here by the group is to compare a group of treated patients with all different types of interferons with a group of non-treated patients. These groups are very rare nowadays, and what has been shown is that you have a higher likelihood to convert into secondary progression by being non-treated. And this is taken often as an evidence that you have a long-term benefit. There has been recently a publication by the British Columbia group in Vancouver that doubted that there is a long-term benefit of interference, which a number of opinion leaders also rebutted by making arguments on methodological difficulties that are related to this study. one being the writer of the editorial, Ludwig Kappos, who is giving the third lecture in the teaching course, who wrote a very nice editorial on what this really could mean. The key opinion nonetheless remains that there is more influence on inflammatory aspects of the disease, means relapse rates, than on real progression. And especially if you start in the progressive phase, then it's not so likely that the interference will impact it. But nonetheless, if you start early enough, then you have, in conjunction, probably also a mild and moderate effect on disease progression. What are factors affecting the treatment benefits? It's pretty clear that this is a protein that you give. It can induce antibodies, and there was a phase while these therapies struggled for who is the best one, that the issue of neutralizing antibodies was cooked extremely hot. Neutralizing antibodies are generated, and they do influence the likelihood to convert into higher EDSS ranges or to have higher rates of relapses. And the rate of antibodies is different. That means the subcutaneous preparations making higher rates of antibodies than the intramuscular preparation, which is being Avonex. But this is only one aspect, because you could also make the argument that the higher frequency subcutaneous interference have higher efficacies than the intramuscular, and that was actually the question back 10 years ago. Who is the most efficacious drug? The head-to-head studies provided some answers, but nowadays one would say, okay, if you look back in time, you could say that higher dose, higher frequency interference have, at least in the beginning, a higher potential of efficacy. Whether this is really translating into long-term benefits with regard to progression, this is not clear. And if you look at studies that try to compare, all have their difficulties with regard to methodology. You don't see really order of magnitude differences in the efficacy, in contrast to comparison like, for example, nowadays with substances as natalizumab or alemtuzumab. This is a big question. Can you optimize benefit? It would definitely help if you could say person A, B, C is a responder to interference, whether D and E is not. This has been a question over the last years and is now being more intensively addressed. I can shorten this by saying, well, we don't have, except for the neutralizing antibodies, not a valid biomarker. IL-17 has been a candidate, highly discussed in the last ECTRIMS in Amsterdam last year, but it couldn't really be confirmed that this is a marker for predicting response to interference, so the levels of interference. Several groups, including ours, we are trying to understand what happens by treatment with interference and maybe therefrom deduce a marker of biological response. None of those is yet at the stage that you could really say, well, a practical treatment decision relies on a biomarker that you could do pre-hoc or post-hoc during the treatment. Secondary progressive MS is a difficult story. I can shorten this up. Interferons have shown efficacy. There have been different trials. One was the European SDMS trial, one was the North American MS trial with different outcomes. And they actually showed, if you summarize this, that the more relapsing you are, the earlier in the secondary progression, the likelier it is that the interferons work, but in general, the order of magnitude of efficacy is not so high. The second substance is a bit briefer, is glatiramer acetate. It works a bit different. It's a polypeptide mixture that, interestingly, was designed as making a supermyelin antigen for experimental studies, a supermyelin basic protein, and this is actually the constituents of it. And the constituents are 4 amino acids that make a composite of myriads of polypeptides, and in general these polypeptides influence immune responses and immune regulation, shifting pro-inflammatory responses into anti-inflammatory responses, upregulating regulatory responses, so a bit different from the mechanism of action of interferons, but in net effect also influencing disease inflammatory activities and maybe also neurodegenerative aspects by providing neuroprotection. That has been a big debate whether glatiramer acetate is directly neuroprotective while, for example, interferons would be not. One key argument were neurotrophic factors that are excreted by GA-responsive cells. Well, what can you say? The treatment trials show that you have an impact of glatiramer acetate on brain atrophy, which is an indirect measure of neuroprotection, not saying whether this is direct or indirect neuroprotection. And disease progression is influenced, at least in the primary, in the relapsing forms, but it is not influenced. And this is a big take-home message. In primary progressive MS and secondary progressive MS, where glatiramer acetate also did studies and they were not positive. So that means if you assume that primary and secondary progressive MS is primarily non-inflammatory degeneration, then at least glatiramer acetate didn't show clinically significant effects. The issue of neuroprotection is a very difficult one. Just to briefly touch this, I mean, it's often argued that drugs would act both inflammatory and neuroprotective, but the evidence to show that is very difficult to achieve because you cannot really extrapolate in vitro studies or animal model studies in what has been shown in clinical experiences, and it's extremely difficult in multiple sclerosis to really dissect direct neuroprotection, which would be, at least by my personal opinion, neuroprotection that is rather independent of inflammatory processes from indirect neuroprotection, which comes if you influence inflammation, and therefore all strong anti-inflammatory drugs would also be indirectly neuroprotective. So the current opinions regarding treatment effects are as such. So you have a natural course of the disease that you influence by intervention moderately, and this is the effect of first-line therapies. This is a statistical effect. I mean, you can have rather full disease control in one patient and only partial in the other. So this is the statistical mean of what happens. If you start earlier, the idea is that you can actually really more significantly influence the overall accumulation of disability, and this actually is something you cannot really regain by starting very late in the disease. And this comes back to this cartoon. What I tried to go through is now the CIS, RRMS, and SPMS evidences for the first-line therapies, and the first-line therapies is the group of A, B, and R, interferons, and C, Copaxone. And to take home for you, nowadays, and I think this is also true with this ECTRIMS, already seeing a lot of candidates coming, also holding the promise to be first-line therapies, is that ABCR drugs are the mainstay of immunotherapy in relapsing forms of MS. Early or immunomodulatory treatment reduces risk of relapses. It influences, at least if you start early enough, possibly and probably also disease progression. The magnitude of effect shows no categorical differences between the different ABCR formulations. At CIS there has been, I didn't go into detail, the concept in the early days that only the high-risk patient would show benefit, that a high-risk patient that has strong inflammatory activity on MRI and residual disease. This is probably not so true. That means that also patients that have lower risks, that means lower inflammatory activities, can benefit from early treatment. But nonetheless, the treatment decision is still an individual one, and so far we have not found any categorical treatment recommendation guidelines to really say, okay, every patient with CIS has to be treated with an immunomodulatory agent, while the percentage of those that are being recommended to do so have definitely increased, at least to our opinion. Thanks for your attention.